Okay, so we should see the record button in progress there. And we'll jump back into the shared screen. Okay, so the plan today, as I mentioned right in the beginning there, is we're now kind of into the specific details of the 3D bioprinter major project. And so I wanted to spend just a little bit of time talking about the confocal inverted microscope, the Nikon microscope that you guys are mounting to, because there are, I think I've talked with a lot of you about it, but there are maybe a few that aren't yet totally comfortable with how the microscope works. And so I wanted to just, give you a little bit more on that. And then I want to jump into the detailed design of a tiny pump. So this is actually an example from fall 2019 that feeds right into what you guys will be doing for the preliminary design report, which is the big kind of major assignment that's out now that you guys should be working on in your group. So so the first thing is confocal inverted microscope. Which looks like this. So you guys have seen a couple of pictures of the microscope. I don't think you've yet seen this one, but the microscope is this instrument here. You guys, I think, have probably seen or worked with a standard microscope before. So it has a lot of features that are similar to a standard microscope, right? It's got the optics, you put your eyes here. It's got a lens system down here. It's got a illumination system and so forth, but it is laser scanning confocal, and it's also inverted. And so what I wanted to talk about is a little bit about sort of how all those features combine together to make this microscope kind of special and unique. But ultimately, this is the one that you guys will be mounting to in the soft matter engineering lab. And you can see it's sitting on, some of you who have done undergrad research will recognize this as a laser table, a vibration table. And so they're very, very concerned about mitigating vibration and making sure that there aren't any vibrations in the microscope as it's actively imaging. And so that feeds back into the customer need that if you do have any motors or anything that's generating vibration, those all have to be off of the microscope stage and effectively off of this table as well. And so you could mount something kind of on this lab bench sitting next to it, or to the wall, or on the shelf up here. But nothing that vibrates can be sitting on either the table or the microscope. And all of this real estate that's on the table, people have asked a couple of questions about, well, can we mount to the table? And when we asked, the customer said no, because they want all of this real estate on the table for all the peripheral equipment that sits there. So, and you can see there's a bunch of stuff sitting on the table. So just to be clear, the 3D printer mounts to the turret, which is this kind of top down mounted device here, or mounted part, which is here. And the way that it mounts to the turret, this picture you guys have. So in this case, in this picture, we've got, I think this is a light source that they've got connected here so that they can do kind of, conventional light microscopy with this thing, but is removable. And when you take it off, all that remains is the turret fixture, which is this guy here. And then this entire section tilts backwards so that you can access it if you wanted to mount something that was different than this light thing onto the microscope. So this is how the 3D printer would actually be mounted, is that the turret, whatever was sitting on there, would be vacated. This whole big support arm, we would push it backwards, and then the 3D printer would sort of come up and interface with the dovetail that's inside the turret. And there are these thumb screws here where you would screw down onto the dovetail that you've built into the 3D printer. And then the 3D printer would be mounted. You would swing the arm back into place, and you then be ready to take your images with the 3D printer in place. And so the customer actually produced for us this kind of close-up image to show where everything goes. And I think you guys have seen this image before. So right here where it looks like there's a light source, this is where the 3D printer goes. So this is, again, the turret. The microscope objectives are down here. So these are when I say objectives, these are the lenses that actually do the imaging. And then the 96 well gel plate kind of sits up on top. And so it's different in its configuration than a normal microscope that you probably dealt with before. For one, because it's inverted, right? So normally you would have the illumination source on the bottom, right? So the lights would be down here and they would shine upwards through whatever your sample was. And then the objective lenses would be on the top. And that's where the collection of the image would take place. For an inverted microscope, it's the opposite. So the light is up on the top and the light shines through and you're collecting images from the bottom. So that's where the term inverted comes from. It's just that the lenses are on the bottom and the light is on the top. And there are a couple of benefits for that or of that. In particular, for our purposes, you can instruments above where you're in, and the image path is still clear, right? So you can 3D print cells from the top and then you can watch them from the bottom. And if you were to do this, sort of in the opposite direction, if you were to try to mount a 3D printer from the bottom and then watch from the top, gravity would be working against you, right? The gel, which is liquid-like, this is the LLS, would sort of pour out of the well plate because it would be sitting upside down. And so that's one of the benefits of the inverted configuration. The other thing that's going on, of course, is that this is a confocal microscope, which is incredibly cool because confocal microscopy takes advantage of a lot of kind of interesting optical properties to allow you to take, and I'll show you guys an image here in a second, but to allow you to take what's effectively a three-dimensional image of your samples. So this is an image that comes right out of Nikon, which is the manufacturer of our, or at least the Soft Matter Engineering Lab's 3D, not 3D microscope, their confocal microscope. And what's hard about this image is that it's inverted. So I wonder if I can just rotate it. Yeah, bing, there we go. Okay, so that's maybe not super clear, but I'll explain here in a second. So right, our optics are on the bottom and our light source is on the top. And the way that I've now got this thing sort of flipped around, and I'll flip it back the other way in a second, but it's now in the same configuration as the inverted confocal microscope that's in the lab, right? With the light source and the sample on the top and then the optics on the bottom. Oops, someone's got a comment. Let's see here. Okay, let's see. So there's a question about, is the turret dovetail connection angled in relation to the plane of the table? Pictures look like it and would require angle on the bracket to support the main 3D printer frame. Is the turret dovetail connection angled in relation to the plane of the table? Okay, hang on. Let's go back here. Let me see if I can get a closer, better view. So I don't know if you're referring to this picture. So in this picture, there's this arm here. So here, maybe let me see if I can annotate the arm that I'm talking about. I have to be careful about my use of colors. Okay, so I'm talking about like this arm, which is sort of cantilevered outwards, right? And this is the turret right here. So in this image here that we're looking at, the arm is actually, how to describe this? It's intentionally sort of pushed backwards. So it has a like a the word is escaping me. Like a hinge. That's the word I'm looking for. It's got a hinge, kind of right here, right? So there's like a hinge underneath, and the arm is pushed intentionally backwards so that you can mount your 3D printer or whatever it is that you want here. But then when you go to actually use the microscope, let me use a different color. Um, this arm rotates around the hinge and snaps back into place, and so it's vertical, and then the dovetail sits out sort of cantilevered above the optics, which are down here. And so if you look at, let me see if I can jump to another image without. Okay, hang on. Let me erase that stuff. So if I jump to another, there we go. Now you could see that that arm is in place now, right? And so the dovetail is sitting out cantilevered above the optics, and the dovetail is, provided everything is calibrated correctly, it is in the same plane as the table underneath, I guess, to address the question. So you guys will not need to worry about like the angle between the dovetail and um, the well sitting on the stage here. They should be in the same, well, they should be parallel to each other, right? So if you design the microscope so that it hangs vertically from the dovetail, it should hang right above where the well plate is, and so you don't have to build in any sort of angle, because the two are meant to be parallel to each other. Does that answer the question? Hopefully. Yeah, cool. Okay. Yeah. Oh, yeah. No, no problem. Yeah, this is particularly hard because you guys can't actually get in there and see the microscope, right? So we're sort of doing this all with like pictures. Um, but yeah, this is a little bit misleading because the arm is actually kind of hinged backwards so that they can get at the dovetail to mount something. Um, okay, so back to confocal microscopy here. So I've intentionally flipped this image over so that the arrangement of the microscope aligns with the arrangement of the figure, but of course it's hard to read upside down. So let me flip it back over. But the point that I want to make is that this image um, might be a little bit confusing because it's actually flipped over in terms of its orientation, with respect to how the optics are aligned with the actual microscope. So, but in general, the way that a confocal microscope works is that there's a laser source, laser excitation source, and the laser, and by the way, the laser is sitting down here, right? So underneath the stage of the confocal microscope. So the laser fires light at a particular wavelength that bounces off what's effectively a two-way mirror, right? So a mirror that will reflect light if it arrives at a particular angle or wavelength, and will pass light if it arrives at a different angle and wavelength. So that laser light is reflected because of its angle and wavelength off of this mirror. It's focused through a set of optics, and then it strikes the sample, right? So this is your 96 well plate with your cancer cells or whatever it is that are sitting on the stage of the 3D printer. And I'll show this in a second, but the laser light does not, at least in this configuration, directly illuminate the cells. Instead, the cells are, I can show you here, they've absorbed some kind of marker material. So we'll talk about this in a little bit, but they've absorbed like a marker chemical, and the marker chemical fluoresces at different wavelengths depending on what it is. And so the laser light comes in at a particular wavelength, and then what comes off is a fluorescence. Fluorescence that is at a series of different possible wavelengths depending on what you've used as your marker. that light that comes off of the cells then goes through the optics again, gets refocused, and at this point goes now through this mirror because it's at the correct angle and the correct wavelength to pass through the mirror. And then here's the really cool confocal part, which is the optics are set up in such a way that most of those light beams don't actually reach the detector, the photomultiplier detector. A lot of them are just stopped um, by a pinhole aperture. But one one point of light or one ray of light actually does go through the aperture, and that ray of light is the one that's collected by the photomultiplier detector, which is a computer system basically that takes those individual photons that are collected and reconstructs the image. So why do this, right? Why block most of the light? Well, it turns out that the optics are set up such that different focal planes in your sample are going to have very slightly different focal points. And all of the paths that lead to a focal point that is not in a focal plane of interest, those all just hit the wall of the aperture and don't go through. And so effectively what you're doing is by blocking all of the light except for the one particular ray of light that comes from the plane that's in focus, is you're taking an image of just a slice of the overall sample. And by changing the optics, what you can do is bring different slices into focus and therefore reconstruct a three-dimensional image, right? So these focal planes are all in the Z direction. And so you just change the optics to adjust the focus of the Z, and you'll be taking different slices of the sample, and so all of the images, all the rays of light that are not in focus just hit against the aperture wall here and don't go through, and then the one that is in focus is the one that passes through and gets detected by the photomultiplier. So what can you do with this? Why is this so cool and so helpful? Well, so normally this image shows, or this slide shows, two different types of things. So normally with a microscope you can only kind of get a one focal plane, right? So you guys have all played with microscopes, I'm sure, in your chemistry and your biology classes. So you basically get just one focal plane. The beauty of a confocal microscope is that you can actually image a three-dimensional volume, right? So not just a plane, but a volume. And with computational help, you can actually enable four-dimensional imaging. So you can image in X, Y, Z, and and so that's what this image here to the right shows. And so I do actually have somewhere, if I can find it, I do have a video microscope. Oh, there it is. Okay. All right, and you guys can't see it because I haven't shared my screen, so give me 1 second. There we go. Okay, so this is that same image, but now I've got the video version of it. So let me just run this a couple times. So what we're looking at is cell division, but image with a confocal microscope, right? So you can see this is actually super cool. All of these cells in the beginning just start out as individual cells, right? And they're in different locations in X, Y, and Z. So different locations in this volume. And all that the confocal microscope is doing is it's just scanning in the Z direction, right? So it's changing the focus in the Z direction. And it's just doing that over time intervals. And then taking those individual images, sort of like an MRI machine, is taking those individual images and like reconstructing them into a three-dimensional volume. And so if I play this, you can actually see, right? So this one multiplied, and then this one multiplied, and then this one multiplies. And then, you can kind of see them moving around and stuff a little bit, but you can actually see what's happening in a volume in the progression of time. So that's what at least one of the capabilities of the soft matter engineering lab, which is super cool. So anyway, wanted to show that. And then the final kind of thing that I want to mention. Oh, there we go. The final thing that I want to mention about this particular technique, I kind of touched on it in this previous slide here, but this idea that you can use different chemical markers to kind of mark either different cell types or different components of individual cells, or organelles, for example, inside individual cells. And with the different chemical markers, once you illuminate them with a laser light at a particular wavelength, they will actually fluoresce with dissimilar wavelengths. So you can see here, I think these are nerve cells actually, and you can see there's three different types of structures. So there's and I appreciate there's and I apologize for this, a red and a green here, and I know some people can't see those, but this was the image that was supplied to me by the customer. So I apologize for that. But there's the blue. I think these are the cell nuclei, and then there's the green color, which is the nerve cells themselves, and then the red is the connective structure between the individual cells. And so each one of those cellular structures has been marked with a different chemical, and those chemicals fluoresce at different wavelengths. And so when the confocal microscope picks them up and reconstructs the image, you can actually see like different structures of individual cells and different types of cells as well, based on how you mark them. So there's an enormous amount of capability here in this instrument to well, here, let's look at this one. There we go. In this instrument, to visualize in 3 dimensions, visualize in time, right? So you can actually watch the progression of cells in time as time moves forward and take a video of what the cells are doing. And with the right chemical markers, you can identify different individual cell types, and sort of watch them and track them and see how they structure and see how they build together and so forth, because they fluoresce differently, and so you can identify them by color when the computer reconstructs the image. And so those are all the capabilities, at least that are important to us in terms of kind of what the microscope is doing behind the scenes for the soft matter engineering lab, and why they're interested in using this particular microscope to add the 3D printer capability to, right? So you can imagine kind of like all of these cool features, right? So you have these potentially different types of cells, right? You mark them with different colors so that they fluoresce differently, or you mark their individual organelles so that they fluoresce differently. And then you can actually 3D print them in a volume, and you can watch in real time as you're 3D printing the cells. And once you're done 3D printing the cells, you can watch those 3-dimensional structures as they evolve over time and kind of watch what the cells do, right? So that's really the motivation for using this particular microscope. And sadly, it's a little bit more complex than maybe the microscopes that you guys have played with in like your chemistry or your biology classes. But it has all these cool features, which is why this is the microscope that we're interfacing our 3D printer onto. Okay, so let me stop there. Ask if anyone has any microscope specific questions. Okay, so sounds like no. So either I've done a really good job explaining, or you guys are all asleep, or you're all really mad at me. Hopefully it's not the latter two. Um, anyway, okay. So let me stop sharing. So I was gonna jump into a quantitative analysis, which kind of leads into the preliminary design report. But given that it's almost 10:30, and I would like to transition into open virtual lab hours, we'll stop here. And I do actually have a buffer day built in this week, and so we can cover this other material, the design of the tiny pump, on Thursday. So if there are no more questions, we'll just stop there, because that's effectively the end of this hour. So we'll stop sharing. Okay, so it's 10:30. it's time for open virtual lab hours. So let me stop recording.